Well, thank you everyone for joining us on what is now our 10th episode of what we've been calling COVID Connect. And as we do at the beginning of every one of these, thank you to the group of community partners here who really got the word out to convene this discussion that's now been going on for the last 9 weeks. You see them up there. No one did this because there was any money attached to it or anything. Everyone just reached out, and one Thursday night we were sending messages out and said, hey, we need to talk about these things. And so many people reached out to their email lists and got this conversation going. So we are very, very thankful. The ground rules, as we normally note, include the webinar will be available. The audio and video recording will be available. The chat box will be available. If I bring you on live, get off of mute and keep it brief. Your name, your company, your needs, your efforts, and your ideas. We do want to hear from you as Michigan and really the country asks the question, how do we reopen? How do we safely reopen? What are the right things to do? And we're going to talk more about that today. Into Being, of course, we do medical device development. So working with FDA and doing development for that is our sweet spot. If there's anybody who's still working on emergency use authorizations or anything like that, feel free to reach out and ask us questions. We are just happy to help you out where we can. And what we're going to do as usual is talk about the latest news. We've got 3 wonderful special guests today. We'll talk a little bit about the status of the emergency use authorizations that FDA has. And then we've got time for Q&A and a community open forum where we continue to want to hear from you. So with that, we want to talk a little bit about the news here that's going on at the moment. So we're going to stop the screen share. And we're going to bring up the statewide, we've been tracking this for a while. This is the statewide bed tracking for the state of Michigan. This may be the last week we talk about this because it hasn't been an issue for a little while, but I think so many of you have enjoyed seeing the actual numbers of the availability of beds in the state of Michigan, as well as the needed PPE that the different health systems need. And it's in pretty good shape right now. But for those of you who are supplying PPE and you're still looking for targets to be able to get PPE to people who need it, the state of Michigan has graciously made that information available from the different health systems. So hopefully that information is useful to you. We talked last week about this COVID Act Now site that has some interesting data, although I'm interested in hearing from one of our panelists today, her thoughts on this infection rate graph, which I just wanted to show you here, at least in the state of Michigan, appears to be coming back up. Maybe we'll get some more information about that from one of our panelists today. But this is an interesting site for tracking some of the stats. Some of the news that we are seeing, I was wondering earlier because the president was in Michigan at Ford, and it made me think, I haven't heard much about the status of Ford's PAPRs and ventilators. We are seeing here that Ford has been shipping PAPRs as well as gowns. We knew they were using their textile ability to get gowns out there. And so they've been doing that as well. I don't know if any of our staff ever did find any updates on the ventilator aspects of Ford. We haven't heard a lot on that end of things, but that's the status of Ford's PAPR effort. And we just had a sort of a breaking thing coming on just before the show. And I'm just gonna read this little bit of news here. But one of our folks, Adrian Harris, had found out that we are now seeing some indications that dialysis machines are actually becoming in short supply. So that's kind of breaking, but we have not heard that this entire time. What's always been the focus was on ventilators and other things like that. But right now there's an indication that there may be some dialysis needs. So we will keep you up to date on that as we learn more about it. But that's the news for today, and we'll talk about the emergency use authorizations a little bit later. What I want to do now is get to our special guests. And the first of those is Dr. Ing Pottinger. She's a virologist who previously served at CDC, Centers for Disease Control, where she developed what's now the global standard test to measure HIV incidence. She's a senior laboratory advisor at ICAP at Columbia University's Mailman School of Public Health. And I want to bring Ing live on the show here to talk to us about the state of diagnostic testing in the U.S. And I hope you're all ready, because I've gotten a preview of what Ing is spending her days thinking about. And it's really, this is like, we're going to go back to college right now and talk about things like sensitivity and specificity. This is just great. Dr. Pottinger, thank you for being on the show.
Thanks for having me.
Oh, that's great. Why don't you bring up your presentation? We would love to learn more from you. A few weeks ago, we had Brandon McNaughton from Acadium on, who was telling us about the sort of the low rate of testing in the US and some of the nuances. You're going to take us up to another level here in terms of the ins and outs of how these tests work and what they can and can't tell us.
Yes, I will happily do that. You are going to have to enable me to share my screen.
Oh, did we miss that one again? Okay, sorry about that. Attempting to do so if one of our staff can see that too. My more button is not letting me do a screen share for you. Why is that? Let's try that.
Great.
Apologies about that.
No worries. Just give me 1 second. No. Sorry. There we go. Are you able to see this?
Yeah, you're showing up great.
Great. Well, thanks for joining, everyone. I am going to take you back a bit in terms of some of the basics around testing, and really talk about some of the updates and what the data really means. So this is a great graph. It was just released in a paper that came out a couple weeks ago, and it really outlines how basically our body's response and how we can measure it when you're infected. So in these early stages, this is the asymptomatic, let's call it window period, where you've been exposed and you start to replicate and make viruses. And up to, it's really about 2 to 5 days before your symptom onset, where we can detect viral RNA from the different sample types, your nasopharyngeal, an oral swab, a throat lavage. These things will start to become positive on a PCR test. So you can see here in the pink and red and blue lines. And once the symptoms begin, again, positive by PCR, and in about 7 to 10 days, you will start to have antibody response. And typically an IgM response is first followed by an IgG, but a number of groups have shown that in fact, these things for this disease are concurrent. And many of the antibody tests, the rapid ones, distinguish between the two, while the ELISA lab-based use combined IgG, IgM signals. And so this lays out pretty well the response and how we can measure them when one is infected. So we're gonna go back to basics here, because the whole point of my presentation is to really help people understand what an antibody test does, what it means when you get a result, and what it doesn't mean, and to understand data quality when picking tests, and how do we pick tests that give us the best data, best positive predictive value when you're telling somebody they're antibody positive. So let's go back to basics and talk about sensitivity and specificity. So the perfect test, which doesn't exist in the world for any disease, the perfect test would tell you 100% of the time someone who's truly negative, and 100% of the time someone who's truly positive with no overlap. That's the perfect test. Yes, you have it, and no, you don't, and it's never wrong. But since that test doesn't exist, we strive our best to hit 100% sensitivity and 100% specificity. And sensitivity is the ability to accurately detect a positive infection, and specificity is the ability to accurately detect a negative infection. So most tests look like this curve, where in blue are the true negatives, and in red are the true positives. And there's always this overlap between false negatives and false positives. And how big this area is depends on where you put the assay cutoff for being positive or negative. And if we have lab folks on the line here, where when you run an ELISA test, or even the RT-PCR where the CT cutoff is, when you shift it one way or the other, you're shifting the balance here between the false negatives and the false positives. So let's just say that this line B is the perfect balance between that. And that's really what you're looking for when you design a test, is the balance between the false negative and false positive. So you don't have more false positives or more false negatives than the other. But what happens if you do shift your cutoff? Let's say you shift it to A. So you've dropped your cutoff. You can see it here. You've accurately called everyone under the red curve positive, but now all of this, which are true negatives, you've now called positive. So your false positives have increased, which means your sensitivity has gone up while your specificity has gone down. Sorry, just bear with me one second here while I move this other thing. And that results in an increase in your negative positive predictive value, because here with everyone that we call negative is truly negative. Whereas our positive predictive value has dropped because now we've included all of these true negatives in our positive outcomes. The reverse is true when you shift, of course, your cutoff to C here. So everyone that we call positive is positive. So you've increased your positive predictive value, but you have increased your false negatives and while dropping your false positive. So you can see this balance that has to happen when you're putting an assay cutoff in there for a qualitative test of being positive or negative. And that's what our antibody tests are telling us. So let me give you an example, and I really want you to understand how sensitivity and specificity, along with positive predictive value and negative predictive value, go hand in hand with disease prevalence. In different prevalence settings, these tests will give you different outcomes. So this is just an example. Let's say we have a test with 95% sensitivity and 95% specificity. And we have a sample size of 200 people that we're testing. And in a 50% prevalence scenario, we have 100 true positives and 100 true negatives. And if we test these 200 people on our test, that's 95% sensitive and 95% specific, it will pick up 95 of the true positives plus 5 false positives for a total of 100, but they're not the true 100. Our positive predictive value in this high prevalence setting will be 95%. Now let's drop it down to 10% prevalence, where we have 20 true positives and 180 true negatives. The test will pick up 19 of the 20 plus 9 false positives. This comes from the negative pool that we've tested. So we now have 28 positives that the test is indicating is positive. Our positive predictive value in this case is around 68%. So you can see the drop that happened between 50% to 10% prevalence. Now let's look at a 1% prevalence, which is probably more, is closer to the scenario we face with COVID in many places in the country. So of the 200, we have two that are true positives, 198 that are true negatives. The test will pick up those two true positives plus 10 false positives from this population, the negative, leaving us with a positive predictive value of only 17%, which means when you're telling someone they're positive with this test in this scenario, they're most likely to be false positive. So sensitivity, specificity, positive predictive value, and prevalence all go hand in hand here in terms of our outcomes. So I know you're going to talk about the FDA and EUA process and status, but there are currently 12 serology tests that have been authorized under FDA. This is 10 of them, and this these are the sensitivities and specificities reported for each of the tests. And you can see here under the sensitivity that our highest number is only 94%, not quite. And we do better with specificity for antibody tests, so they are pretty high on this side. And you can see the Wadsworth Center, the New York State lab, they set their cutoff a bit differently intentionally, so their sensitivity is around 58%. And
And we're gonna do questions afterwards, but as we were talking earlier, and your slide makes it very, very interesting there. I just want to ask you one quick question.
yeah,
Why
please.
are, I think our audience may not understand, why are the sensitivity and specificity so different per test? Some of us who have done
Right.
molecular diagnostics have a clue, but can you talk for just a minute about why those numbers are so different? Because I think a lot of people go, well, it should just be a test. It's just one binary thing. And you've already pointed out a lot of that, but why are even these numbers so different?
So it depends on your target, what protein is used for detection.
Hmm.
All of these, like they're detecting antibodies, IgG, IgM, but what are the targets for those antibodies? So some of them are focused on the spike protein, some of them are focused on the N protein, some of them are specific to like a certain binding site. So it depends on the makeup of the test and the antigen target, as well as some of the characteristics in the development and where the cutoff is. And so especially like, let's say they target the N protein. Well, the N protein has a lot of overlap with other coronaviruses, and that affects the sensitivity as well, and it will affect different populations. And I gave this example previously where if you test in a Hong Kong population where they saw the SARS-1 outbreak, some people might have some cross-reactivity because some of these proteins are homologous. So those are all the factors that go into this, right? The design, the antigen, the protein, the way that the system detects the antibodies. So it's a whole host of things, and every manufacturer is going to make a different test. So none of these things are consistent between these. Like the Mount Sinai laboratory, they have their own in-house protein that they use, and they use two different ones for their assay. So those are all the factors that go into understanding. This is why you cannot assume a test is a test. And I know a lot of Americans really think that because, you just send a sample off to the lab and the lab gives you a result, and no one ever questions the quality of that lab test, because the lab is supposed to be this perfect place where you get perfect results. But that's not what happens. And it's all based on the quality of the test, and obviously the quality of the sample and all those things, but it starts with the test itself. And that's why the validation data that the FDA now requires is so important. And we are still waiting for the FDA's data, where NCI, they asked the National Cancer Institute to do test-to-test comparison. I believe it's been 8 weeks since they began this. The data was turned over from NCI to FDA, and they still have yet to release this. And I am very interested in seeing that data. There's a separate group called FIND. They've done an independent evaluation of the RT-PCR tests that are available worldwide, and now they're doing an independent evaluation of the different antibody tests as well, but that data is not available yet. UCSF has produced some data, and it shows these differences. So it's very important to understand the sensitivity and specificity, plus the population you're testing when selecting test kits. So, right, you're not going to want a low sensitivity test in a very low prevalent setting, because all of your data is probably not reliable.
That's great.
Yeah.
Just to understand how those things are so different, and how many variables there are that go into each of those is really, I think, helpful for our audience. That's great. Okay.
So this is the New York Wadsworth assay, and this is the data that they submitted to FDA for their EUA. And the point to this is what I want to make is about the timing of when the samples are collected. So if you have someone who just showed symptoms, they tested positive on PCR, and then you put them on an antibody test and they maybe come up negative. Well, why would that be? It's because your body hasn't made enough antibodies to be detected. And so you can see here in their data that over time the sensitivity increases, and that has to do with the biological response. So the farther out from your symptoms you are, and if you're suspected of having case, the more likely that result is reliable. But if you're taking a sample, within a week of being having symptoms and coughs and all these things, your antibody test may not come back to be what you think it would be, which is positive. So there's a timing component in this as well. So how do you improve your accuracy? And this is the most important thing. You can see that in our low prevalence setting. And the reason why I think we are definitely in this 1 to 5% prevalence, and 5% is really me being conservative in terms of like how high it might be. New York City is a different situation, but in the rest of America, and especially outside of large cities, I'd be shocked if our prevalence was above 1-2%. Even if you do the math on the numbers right now, I can't imagine that our national prevalence is going to be higher than 5%, and 5% is a long shot. And this is accounting for all of the asymptomatics and the people who never got tested, but who might have been infected. But in terms of serology testing, because everyone keeps talking about serology testing, and all of these doctors are offering tests. You can go into your dentist's office or the dermatology office, and for a few $100, they'll give you a test, right? But what if you get a result back and it's positive? Can you believe it? So I really want, especially people, to understand if they're going to offer these things, to do it in a way that is responsible and gives people good quality data. So what's the best way? Use 2 tests. If you use 2 tests in an algorithm format, and we do more than 1 test for HIV. We do it for many, many other diseases. So it shouldn't be, there's no one test that's going to work for COVID. So we need to use what we know from other diseases to apply it here. And, I'm going to give you the HIV example in the next slide, but, pick a really high sensitive test for your first screening test, as close to 100% as you can get, and then pick a really high specific test so that you can sort out the negatives and the positives. Let me give you an example of why that order is very important. So let's say you have a test here. Let's stick with the sample size of 200 people being tested. And now we have a test that is 99% sensitive and 95% specific. So of those 200, 99 true positives, sorry, this is still, let's keep with the 50% prevalence. 99 true positives detected plus 5 false positives. So our positive predictive value in this case is 95.2%. And the percent false positives is the 5, so it's about 4.8. Now, if you take a second test and you run them on those 104 samples, it will screen out those 5 false positives and help you clean up your data. So that improves your positive predictive value from 95% to 100%. We see this. I'm going to show you large scale numbers of how well this works when you do it in an algorithm. So this data, you'll have to bear with me. This is real data, and it's from hundreds of thousands of people that we've collected all over Africa as a part of our HIV programs that Columbia does for the US government under the PEPFAR initiative. And we did surveys in 14 countries, and in each country, we use their national HIV testing algorithm that's it's a rapid test, the rapid test algorithms. And in this case, HIV tests are the best tests of any disease in the world. They have high requirements for approval both by WHO, FDA, everybody. They have to have a sensitivity that's greater than 99% and a specificity that's greater than 98% just to be approved. And most of them perform better than that. But we still, because of the sheer numbers of people that get tested in our programs, we still have many inaccurate results. But we overcome some of that by doing 2 tests. And I gave 2 examples here. This is the Determine HIV rapid test. Some folks might recognize it. It is the most widely used HIV rapid test in the entire world, followed by Unigold here. So I put each row here, the orange, blue, and green represents data from one country. And in green is the HIV prevalence. So I put them in ascending order for you. So you can see it spans down here from 1.4% all the way up. And then what the positive predictive value when we only used 1 test versus the national algorithm of the 2 tests. So in a low prevalence setting, you can see with just 1.4% HIV, the positive predictive value of 1 test was 55% roughly. But when we added 2 tests, my arrow keeps disappearing, it increases to above 90. And then you can see over the different prevalences that with 2 tests, we're near 100% every time, regardless of how well the first test works. So this is really trying to drive home this point that you can do better by just doing a second test. You get better positive predictive value because you don't want people to believe wrongly that they have been previously exposed and have immunity and go out and go, sing at choirs or go to bars and seed a large number of people and because they thought that they were immune to getting reinfected. So it's really important, this data quality piece, I'm a big preacher of it, that one test is not reliable, especially in these low prevalence settings of COVID. Really work on doing a confirmatory test if you can. And so these are just some of my thoughts on this about, I mentioned how FDA and NIH and NCI and CDC are doing this test kit evaluation, waiting for that data so that we can see how these tests perform. Of course, a biorepository is helpful for kit developers and for people to do their independent evaluations. And different testing algorithms, I think our federal agencies need to provide some guidance to people on how to use the antibody test. Because right now, those guidance documents are not very helpful, in my opinion. But that doesn't mean programs can't make their own decisions in terms of being informed about test data quality. So do 2 tests and make sure that, especially if you're doing a survey and you want to know what the prevalence is in, the city of Detroit, then make sure your testing population is representative. And I think, Jean, you and I have talked about what is representative and how to extrapolate data. So, right, you wouldn't take a PRISM population and extrapolate it to the general population of Michigan. Those aren't equivalent. And, if you're interested in gathering more data on your testing, collect more samples from people. I mean, people are generally giving in this period, right? Like we see all these people who are willing to donate plasma for the convalescent plasma programs so that we can get more data behind the testing quality. And this will help you, especially this disease isn't going anywhere. All we can do is get better data so that we can better manage what we produce. And then, of course, understanding the limitations that even with two tests, there's still going to be some false positives in there. The same way we have, there are limitations to the RT-PCR tests, to the diagnostic ones, but that was not the point today. But all tests have limitations. There's no perfect test out there for any disease. So really knowing what we can and can't do and what a negative and a positive result really means. And I'll stop there.
That's phenomenal. I think one thing that I finally got, and I've had the privilege to sort of hear a version of this presentation now a few times. You and I first met when Stacy Frankovich put us all together with the MedHealth cluster, and we really enjoyed that presentation. And I think what I finally just realized, as you said today, that the false positives are sort of one of the dangers of this. And you think to yourself, at some level, oh, why is that bad? If I really didn't have it, then I'm okay. But I think the point that I'm starting to see there, correct me if I'm wrong, is that if we're seeing false positives, then at this point in time, people are going to say, I've already had it, I'm fine. I can't infect anybody. And that's obviously exactly not true at that point.
And your behavior changes, right? Like imagine if I found out right now I was antibody positive. You might go to the store without a mask on. You might go to a bar and get a drink, right? Like a little nudge here, a little nudge there to be like, well, I'm okay. I've had it. They say I can't get reinfected. So people's behavior changes when they find out they're antibody positive. And we actually don't know how much protective immunity lasts. I do think there is protective immunity. There's no data. The disease hasn't been around long enough for us to understand. Is it 6 months? Is it a year? So, really to message that to people who get positive results and what it means.
Yeah, that has become very clear to me as you've presented this. That's awesome. I see a couple of questions here from Olaf Ile. in the chat. And also anyone else, if you've got a question for Dr. Pottinger, feel free to raise your hand in the Zoom room and we will we'll bring you on. But from Olaf here, we've got a couple questions. One says, Dr. Pottinger, the listed 10 antibody tests, are they all specific to SARS-CoV-2 virus-driven antibodies, or are those general antibodies fighting any kind of infection? I guess the former, but maybe not as specific, e.g. all coronavirus. I think you sort of addressed that, but maybe you can just touch on that specifically.
So they are specific to this virus. Some of the tests do include the N protein, which is very homologous to the SARS-1, it's about 70%. So some cross-reactivity, but as you know, when you submit to FDA for EUA, you have to, in fact, test the 4 human coronaviruses that circulate every year that cause colds. About 15% of colds are caused by the other 4 coronaviruses, along with the SARS-1. And almost all of these, I haven't seen a single test where they've shown cross-reactivity to the other coronaviruses that we know about.
Great. The other question here was, he said, final thoughts and suggestions, and answer this as insofar as you think you can. Who would be the best organization to provide this guidance and implement them? Is it each state, county, city, or should this not be available from CDC? It would be guidance specific to prevalence range from low to high. I think this is alluding to your different areas have different numbers, i.e. does CDC offer that template? Again, only insofar as you think it's within your domain.
Yeah, so I think that CDC should in fact be the right federal agency to provide the guidance to different states and localities to do their surveillance. They do this for other things, so it's not, that's within their purview and outside of FDA and NIH's purview. So they need to do that. The guidance that's available now is not specific at all because they don't want to endorse a certain test, but they do need to provide guidance on the algorithm use. They are aware of this, as is the National White House Task Force. I've had conversations about, the need for guidance on testing algorithms with them. So it's just a slow moving process. And hopefully that guidance will come out. We're starting I know that their international task force within CDC as well is working up guidance for our international partners. We work, CDC works in all of these other places. They need to provide guidance to countries that they support as well. So, and that is all under the US government, work under our PEPFAR and our CDC programs.
Good. From Wendy Bloomberg, what is the gold standard reference used when determining the sensitivity and specificity of the tests?
Yeah, so the gold standard right now that people are submitting for EUA are RT-PCR positive, confirmed positives, and hopefully collected within the right time frame. Obviously I said earlier, if you're collecting it, when you're collecting that nasal sample, it's not gonna give you the right result. But I think most people do understand that, most of the test kit developers. But they are all RT-PCR positive, and there's no assumption in their positivity. They are confirmed positive by PCR.
That's good. That's great. I'm glad for the question. Glad for the answer. That was good. Any other questions? Please feel free to raise your hand in the Zoom meeting. I guess one question that I had, Ying, was in terms of those numbers that we're seeing, and I think I first became aware of this when I was looking at some of the CDC numbers, and they were showing things. Again, it's not the same thing because deaths and infections have very different. I'm not trying to equate those two at all, but the CDC was looking at excess deaths, and you see some very, very different numbers all over the country. Some are just incredibly high, and some are basically normal. Is there anything that we can say about why, or is it too early? Why do we see New York being as, I think the last I saw was they're seeing numbers that are like 230% of normal. I mean, it's these incredibly high numbers, and you get other places where they're not high. And so people don't have uniform impressions about what we're up against, which is unfortunate because it's everywhere, but it's having different effects. If any idea why that is, there anything we can say about why that is? Why are we seeing such crazy different numbers?
I think some of it has to do with reporting issues. I think it has to do with there are some places where when there are deaths, especially like in nursing homes and stuff, where people are dying from the disease, but they're not being tested and counted.
But I should be clear, I'm not necessarily speaking to deaths so much as just like the infection rate that you were talking about earlier with respect to your infection.
The attack rate that or
Yeah.
prevalence in different places. So far we're counting people based on their RT-PCR, and we're starting to get data out of California and New York about prevalence, that they've done antibody tests in a more, convenient sample. And that number in California has ranged from about 2.5% to 5.5%. And then in New York, they sample people going to the grocery store and found a 21% prevalence amongst that population. So the first thing for me is to say that number is probably higher than it really is because of the testing quality, right? So the 21%, it's probably really like 15-16% in New York City in that population. So that is a convenient sample. I wouldn't project that out to everybody unless they designed a study that was more inclusive of what the population in New York City looks like, right? Like if you do a surveillance study, it needs to model what your population is.
But it does sound like, and correct me if I'm wrong, it sounds like you're seeing enough to say, even if the numbers are wrong and will be corrected,
yeah,
you are seeing that maybe California does have a lower infection rate. I don't want to make things on faulty data. That is like something we can say.
Yeah. That's definitely true. And that's just with the sheer numbers that we've seen tested positive, right? The positivity rate in New York City is the highest in the entire world. And that's true. It's real. That's real data. And, there are a lot of things, there are a lot of factors that go into both the prevalence, the number of people positive in that population, and the number of people dying who are infected. New York City is a special place, right? Like everyone's on top of each other, a slower response on the front end. People continue to use subways. It's super, super, super dense. I mean, I lived there for years. And New York City folks are a little bit stubborn. They're like, well, I'm gonna take the subway, right? Like that's just, it's density has a huge piece to play in all of this, obviously. Whereas, I grew up in Saskatchewan. Saskatchewan doesn't have, in Canada, right? Like, and they don't have the numbers that you see in New York City because people socially distance because that's normal. It's just not as dense there. So they don't interact with each other in the same way to allow the infection transmission to occur.
It's interesting because we were, I think it was maybe that same phone call that we were on, we heard from UCLA and the relatively low numbers despite the very high population. And of course, the difference there would come down to density, would at least be one of the factors to consider.
I mean, it's dense in LA. I live there too. But folks spend a lot of time in cars. You have to drive. It's over a larger area. So there are a lot of people there, but you're not on top of each other in the same way. You don't rely on, metros and stuff like that to get around. So it's very different. And yeah.
I want to get to two more quick questions here before we're out of time. But Dan Hathorn said, if a health system is only using one type of PCR and one type of antibody test, does using the same tests in multiples still bring your PPV closer to accuracy needed to eliminate false positives and false negatives?
So no.
Sorry, Dan.
Sorry, Dan. Your PCR tests are working, like the ones that we have access to here in the US, with the Roche and the Cepheid and the Abbotts, they work really well. If we were using the China CDC kit, I would be very concerned and be telling you a different story. In terms of the diagnostic piece, there is some data right now about the Abbott ID Now not working as well as originally thought. And so the company is undergoing some data review. Folks have reported it out to FDA. And I would say that's the only one. I think the best performing test is the Gene Expert, the Cepheid piece. It's the data from all sorts of groups have shown that that kit is working really well. But the antibody test, so if you do it twice, this is like of HIV viral. If you do one sample twice, you're going to get two different numbers. That's just the variability of the assay and the sample. But you're not going to improve your positive predictive value if you run the test twice. You will improve your precision, but not your accuracy.
Great. Good answer. Yep. Okay. The other question, I don't know if you have any thoughts on this. One of the discussions that we've been having is, as you look at numbers, there's the whole question of mortality, which is some of the first things that people were looking at, but then the question of morbidity, and not just of SARS-CoV-2 seems to work on comorbidities as one avenue, but the question of, does it produce morbidities that wouldn't have been there? And so as we go forward and as we look at its effect, and I know your focus isn't necessarily as a clinician, but if you think about morbidities, do you have any initial thoughts or what you've heard in terms of how it affects, and is this something that temporarily affects people, or is this something that has the potential to affect people in long-term ways?
Yeah, so with SARS-1, people were shocked at how much it destroyed internal organs for a long time. And then it wasn't helped either by the initial clinical response, which was to give people super high doses of cortisone, right? So then it weakened people's bones and all sorts of things. I think we've come a long way from that experience to this virus now, but there's so much we still don't know, right? Like we've passed the 5 million mark. That's likely an undercount, but we're still finding out new things that this virus is causing, like the Kawasaki-like disease in children that they didn't report in Italy or in China, until recently. So there's still so much to know, and it behaves differently in different people because obviously we're not all the same, right? We're all facing different, like, populations in our resource limited countries face different infections that they battle, right? So their immune systems are different than our immune system. We've shown data in my lab that IgG levels vary, just basic total IgG levels vary by different populations because we all face different things, and in general health. So we are learning still, and there's nothing predictive so far in terms of the data. We just, and the thing is, with the 5 million people infected, we don't follow every single one of them, right? So we can't collect as much data. I know there are a lot of doctors out there who are very research oriented, who produce and publish this data for other clinicians to know, but certainly the majority of cases don't get written up about.
Certainly something people are going to be studying for years to come. I do have one hand raised, and that is from our friend Stacy Frankovich, who was actually the one who introduced us. So I got to let Stacy on here just to say a word.
Thank you, Jean. Hi, Dr. Pottinger. I'm so glad that you were able to come back on and do this.
My pleasure.
Really quick question. Is it possible for this virus to adapt? And by that, to change the way it interacts with us over the course of time?
Yes. So the answer to that is yes. All viruses, this is how viruses are very, very clever. They adapt, and part of their machinery allows for mutations. The human, when we replicate our DNA, we do it with high fidelity. We call it high fidelity. Very few mutations, but when viruses replicate their genetic material, they allow mutations in. It's part of their replication process, and it allows it to adapt. That's why HIV is so clever. We don't have a vaccine to it. And this virus, there's a great website out there, I'm happy to share it afterwards, that people have submitted full-length sequence data to that track the changes of the virus since it emerged in China. And you can see different lineages coming out of it. Now, so far, these mutations have not resulted in increased infectivity, at least that we're really seeing. There is a report out from the Los Alamos lab, which is a top-notch lab, warning that there is possible adaptation happening, and it could result in a more infectious, a more fit virus. And then there's also other data that I've seen where the virus is being attenuated through various deletions. So it is adapting to our population. Because it's infecting so many people, it's able to keep adapting, right? Like if it only infected 100 people, it wouldn't have as many opportunities to mutate. But because we're at 5 million, it has many opportunities to mutate over time. So it's something that people are watching very, very, very closely to see if, especially if there are any adaptations in the way the virus infects. So that binding domain to the spike protein, whether or not it is, makes it more likely to infect a cell or not. But yes.
Great. Well, thank you for that question, Stacy. And if you're able to, if you'd like to share that website, if you were just to type that into the
Sure.
all panelists and all attendees chat box, I'm sure people would love to follow up on that, because that's a great
Yeah.
resource.
It's a great website, and if you play it, it will show you the changes and mutations over time and by geography.
Dr. Pottinger, Columbia University, thank you so much for spending this time with us today.
Thanks for having me.
Really appreciate it. Have a great day.
Thanks, you too.
That was phenomenal. I been waiting for that kind of information, so I just really appreciate Dr. Pottinger's participation. Next up, I want to bring Jerry Douglas on for just a couple minutes. Jerry, you can unmute your mic and your video. Jerry has been a regular attender of our COVID Connect webinars since I think day one. And Jerry has been working behind the scenes on mask materials. Of course, it's something that he's been doing for any number of years. I think he told me it was 13. I'm trying to help you start your video there, Jerry, but I'm having a little issue, a tech issue on my end. Maybe you can start your own video there.
Okay. All right. Is there anything I need to do on my end here?
Just go down to the bottom and say start video. Will that allow you to do that?
It says unable to start video. You can't start your video.
Oh, great. We're having one of those bad tech days.
It's because you're the one that's stopping it. So.
Oh no. Let's see here. I don't know if one of my if Steve behind the scenes can get Jerry's video started. Feel free to.
It might be better anyway, since I haven't had a haircut in the last 6 weeks. So that's why I'm wearing a hat, actually. So
very good, Jerry. I do apologize. If Steve can get that started for us, I'm gonna have him do that. But my system is actually not responding to the mouse click to get that to happen. I'm glad that the webinar is still running. That's a good thing. I wanted to. Let's see, Steve. I can't do that either. Okay, we're just going to keep rolling here. I apologize for this, everybody. Try that one more time. Did that give you the option, Jerry?
No, it still doesn't. It says unable to start still, but anyway.
Okay. We'll keep working on that here. But anyways, I wanted to let everybody know, and I do apologize for this. Jerry and I were talking earlier this week, and Jerry is with Innovative Air Management, and you had been sending material samples almost from day one to a number of folks working on these efforts. Mm-hmm. And when you and I talked yesterday, I think it became very clear to me that while your sources from your material are in China, you guys have been doing a lot of US-based testing of those to
Yes.
some of the standards that we're all concerned about for N95s. I'd love to have you just tell folks a little bit. I'm sorry, the video's not working, but I'd love to have you just tell folks a little bit more about what you've been working on.
Well, we actually have been working on this for a couple of years to come up with an alternative to some of the other HEPA-based medias for the mask, as well as other industries like gas turbine and other other places where they're using a lot of the HEPA grade filter medias. But so in what we have actually done is come up with a PTFE membrane, and that's what we do. We're one of the world's largest manufacturers of HEPA grade PTFE. And so we have this this filter media that we actually manufacture in China, and I'm a part of that development of the different membranes. But then we bring them over to the US, and then we have everything third-party tested. And this mask media that we offer has been tested according to the CFR 42 Part 84 test. And so we have multiple tests at different micron sizes. We even have what we call a HEPA testing, which gives us the full spectrum of the particulate or the efficiency at all a full range of particle sizes.
That's really cool. And you shared with me one of the test things that it's done by your lab in Kentucky, and they're certainly targeting some of the right stuff there. That won't resolve all the issues that a mask manufacturer would have to do, but certainly give them a good leg up on it. which I thought was cool. One of the things you and I talked about the other day was that you are, your factory in China is at least FDA registered. So you guys are at least known to FDA,
Yes.
and that's for, what is that for? That's for the PTFE dental floss, I think, right? That you guys.
Yes, we have FDA registration for the dental floss, and we also have it for our stable fibers. So there are actual fibers and stuff that go into the raw materials have the FDA certificate. And then we will have the FDA certificate on our mask media. We were told that we should have that Tuesday or Wednesday of next week.
That's great.
That's the thing.
Well, we certainly appreciate you having participated in all these webinars, and thought that we'd like to hear from you about what you've been up to. What has been happening? What's been happening behind the scenes as you've been trying to supply these materials?
Well, we were on your webinar, or a part of it, and I asked a few questions, and from that, I got a barrage of phone calls from people all over saying, hey, we want samples of your media. And luckily I had samples here. And so I sent samples. My FedEx bill is well over several $1,000s.
Oh my goodness.
But we've sent samples all over the United States. Hawaii, we're actually sending a lot of filter media to Hawaii, the government's Hawaii. We started, DOD. A lot of the clothing manufacturers. Everybody's buying these machines and making their own mask, and which is awesome. So everything's being done right here in the United States. And so we're one of the major suppliers in China of the filter material that they're using. We're probably making somewhere around 5 to 6 million masks a day with our filter media. And so now I brought the media over here, and I'm in the process of making it here in the United States. But it's a very lightweight media, which means that it's going to be great for the summertime, because it doesn't retain all of the heat and the moisture. It's 100% hydrophobic, which means it's 100% waterproof. And the biggest thing is that we have a high capacity. We were able to produce for the United States over 5.5 million square feet per month of this material. And we store it in our Michigan warehouse right here in the Detroit. Actually, it's at the Detroit airport. And we also have it in Toronto. So
that's wonderful. Well, we really appreciate what you're doing. And how can folks get in touch with you if they're interested, if they're making masks and they want materials?
Well, they can either call me, and I don't know if you have the phone number in your database.
I think we still have it in the contact list from previously, so they can get it there. Maybe if you're able to just type it into the chat box.
Okay, I will.
That would be
The other way is through our website is Innovative Air Management, or innovativerairmgt.com is another way. That's like I said, we've been in business supplying the United States, and especially Michigan, with media for the last 7 years. But we also have brought in masks too. So since we have a strong relationship with our customers in China, that they give us the mask in large quantities, and so we're bringing over the what we call, I guess we're just calling them safety masks, to help people, the stores and factories, to be get their people up and running.
That's wonderful. Well, Jerry, I really appreciate your coming on with us today and talking with us about this, and kudos on the good work. And we do hope that we continue to get connected to folks. So thanks for being with us.
All right, thank you.
Appreciate that. Well, we are just having a tech meltdown over here with Zoom. A few things have changed, and I am worried that my video of allowing other people on video has actually stopped. So Stephen Rapundalo from MichBio, President and CEO of MichBio. I don't know if your video is going to work or not, but you're welcome to try to turn that on if you can. You can do that right now.
Well, Gene, can you hear me?
I can hear you just fine.
The system's telling me unable to start the video.
Oh, I'm so sorry about this. This is really weird.
You stopped it.
Yeah, that's what it's telling, and I am clicking on the more button, and it's not letting me in. So, oops. Now it's okay. There we go. Zoom is melting down. Well, Stephen, we're gonna have to do this without video. I do apologize for that. But thank you for hopping on relatively short notice today. Just to tell our audience, I reached out to Stephen because MichBio had put out there about the state of Michigan's opening up of research labs, and I said, oh well, Stephen, if you've been thinking about this, you should come on and tell us about it. And not only has MichBio been thinking about it, but you guys have been intimately involved in that conversation, which I hadn't realized until recently. So I wanted to turn it over to you to tell us about the research lab reopening and what you guys have been doing behind the scenes.
Yes, well, well, first of all, thanks for having me again. Always a pleasure. Yeah, the whole situation with the lab reopening evolved very, very quickly over literally a matter of hours. Last Friday, the governor was planning, I think my understanding is at the urging of the research universities who have been anxious to restart lab operations, was intent on issuing an executive order to provide guidance on reopening labs. And the proposals that had been made by the universities, I guess, they failed to consider implications for industry until the very last minute. And so just right before noon, I got a text from somebody in Lansing who had spoken with the governor and said, well, let me get back to you. We can kind of get a quick read from industry. And thankfully that happened because the restrictions were a little, well, they were too restrictive on a number of different levels. Particularly, you have to, I think everybody has to be mindful that we were an essential industry, and so there we go. Most lab and certainly manufacturing operations had continued throughout this stay-at-home emergency order. And so companies had already put in place various measures to contend with social distancing and so forth. And so I think the realization came that they needed to sort of comport with what was being, whether what industry was doing comported with what was being proposed by the universities.
And we had this sort of situation where companies were sort of maintaining minimum viable operations, and the universities were pretty well shut down. Now the universities are opening up, but they're proposing some things that perhaps are more restrictive than the companies were ready to do. So we're seeing this sort of disjoint. Is that fair?
Exactly. And so there was three major concerns that we had. First, foremost was what was being proposed for space restrictions. The initial proposal was one body per 144 square feet, and which was double essentially the social distancing. And we pushed back pretty hard on that. And so what it ended up in the executive order was that each organization could work it out with MIOSHA on a case-by-case basis. The other issue related to temperature checks, basically doing health screening of employees. And the temperature checks were proposed to be mandatory. We pushed back both from a clinical utility standpoint as well as from a practical one. I mean, for the first hour after the call that I received, I immediately sent out texts and made calls to CEOs of large and small organizations, to incubators and so forth, to get as diverse a read as possible. And certainly got a lot of feedback on what was practical and what wasn't. With the temperature checks, as you can imagine, the larger employers like Perrigo and Pfizer and Endo Pharmaceutical, when you've got hundreds of employees coming in for a shift, to do a temperature check on each and every one of them obviously isn't practical. So that was watered down as well.
We've got that up on the screen right now. It's that clause right there with, if possible, a temperature screening, which sounds like is what you guys were working on.
And then another related issue to temperature screening was concerns over data privacy. What was going to be done with that data? How, what kind of liability issues did it raise for companies because they now had access to employee data, and so that too was kind of we pushed back on and was a little bit more made more general to provide more flexibility. So the governor wanted to issue that at 3 o'clock in the afternoon. She ultimately issued it about 6:30 for last Friday evening. So it was kind of a whirlwind period of time between the initial call that I received and trying to get a hold of all the principals at companies all over the state. And I'm grateful that there wasn't anybody who didn't respond almost immediately to the urgency to get this information and get the feedback back to the governor's office. So
that's really, really phenomenal. And yeah, like I said, I just hoped for an analysis of it. But then when you and I talked, you guys had been working feverishly, which is, with so many things, is you guys are often working behind the scenes on these things where the concerns of our biosciences industry touch.
Well, when something
politics.
like this comes, when something like this comes along, you pretty much drop everything else and
run into it.
I was in the middle of another teleconference. I was basically multitasking, and this obviously was critical. We didn't want to have our organizations impacted detrimentally because of not having had sufficient input.
I was really glad that you brought up to me the whole question of the privacy protection, because it's something that didn't initially cross my mind, because of course FDA's perspective with respect to thermographic systems, something that we've looked at, is that they are medical devices. So what we're doing when we take somebody's temperature looking for fever is we're taking a medical, otherwise HIPAA controlled record about that person. And so now it's that we're always interested in these sort of privacy concerns, but now your employer has this otherwise HIPAA data about you on a day-to-day basis. And I'm glad you guys were thinking about that.
Well, that's an issue I think that's been certainly widely discussed nationally. It's a critical issue because, what can the data be used somehow by an employer to impact employment? There's all sorts of scenarios, legal scenarios that one can conjure up. And so we wanted to be sure that there was some flexibility in there so that both the employer and the employee, had some assurances that that they would, both their sets of interests would be protected.
Well, that's wonderful. We appreciate that update and all that you guys have been doing. As long as I got you on here, Stephen, anything, I know MichBio is doing all sorts of stuff. Anything you want to tell us about real briefly right now?
We've had a series of town halls with some of our federal elected officials. We're trying to book some other ones. We've just, I think are announcing today that we've got the first town hall with state legislators geared up for next Friday morning. We're going to be doing some briefings for legislators on a variety of topics. You had your earlier guest talking about COVID testing. That one is in the works. We've got one coming up in a couple of weeks that we'll be announcing early next week. That'll focus on convalescent plasma therapy. A number of our companies are working in that space, and we want legislators to be educated on what all that means. I would just say check out our events calendar. There's a whole myriad of webcasts on a range of topics that are coming up.
Well, we appreciate that and all you do, and thank you for this update and all the work behind the scenes, Stephen. Really appreciate it, sir.
You're quite welcome. Have a great weekend.
Thanks, Eric. Thank you very much, and thanks for being with us. Looks like we got the technical issues resolved there. Had to go through a different window, but I saw Nikki Kennedy's note there on the topic of temperature screening. The accuracy of these at-the-door measurements is incredibly low, something to be considered when using that metric to base someone's health upon. And I think a lot of us have heard of those anecdotes where temperature screenings were used sort of prematurely before people thought about the different temperatures that a human body takes depending on where it's been immediately prior. Stories of people getting kept out of businesses or work because they'd sat in a warm car. So then we know that there are people working on those kinds of things. So hopefully that will get resolved as time passes. Real quick update on the EUA status. I think Dr. Pottinger alluded to it earlier, but right now there are 76 test kit manufacturers and commercial laboratories with assays. There are 29 high complexity tests. Two new things that have popped up on the EUA front. The EUA front's been relatively quiet this week with respect to a lot of the things we were initially talking about, but I want to share two things with you. One is this, the Walter Reed National Military Medical Center got an EUA for a system that actually I'd be interested to hear from our friends at MCIRC at some point. Sounds an awful lot like the MCIRC system, but it's basically a negative pressure device that includes a bag and some framing to put oxygen in and then draw vacuum out. It's called the CAMIC system, and it's to prevent healthcare professional exposure to pathogenic biological airborne particulates as an adjunct to PPE. So you're wearing your mask, you're wearing your face shield, but then you're also doing this, and it's a large clear plastic bag over a box-like frame, and it isolates, and then there is a flow to the patient's nose and mouth of medical air, which is then sucked out by a vacuum on the other side. So that's, Walter Reed just got that EUA. Steris got an EUA for N95 decontamination, and this based on steam decontamination, which was fascinating to me because as we've been looking at N95 mask decontamination, steam seems to be about the worst thing you could possibly put on an N95, but we're hoping and trusting that they've validated that. But this is another decon system that's out there, really a cycle, because a lot of larger hospitals have these systems in place, and they are using them now under these approved cycles. Want to give you a quick highlight on Appendix B, the ventilators. There's really only 2 new ventilators this week. And so now we've seen a lot of these Chinese-made ventilators make their way onto the EUA list. It's a pretty long list at this point, folks. I didn't actually count the number on here, but I'm scrolling up and scrolling up. And if you're still seeing my screen, oops, I just went back there. It goes a long ways up. So there's a lot of ventilators on the list at this point. So that is the update on the ventilators. What we want to do right now is we want to switch over to Q&A as well as any open forum things. So first of all, Q&A. If anyone has any questions that you would like to ask about the things that we've talked about today, or really anytime on the show, we would be happy to address those now. I'm trying to bring up the participants list. Raise your hand if you've got a question. And if I don't see any questions, we're going to go right to open forum and hear from anybody who's got some information you want to share about your needs, your efforts, or anything else that you're working on. Maybe you've got an idea that you want to get out there. We'd love to hear from you. First one I see here is Ken Spencer. Ken, you're live.
Hi, Gene. So in regard to the Walter Reed EUA thing there, that's based on a company actually out of California, Rapundalo. And it's primarily for transport of patients. If you go on their site, you can see what it looks like. It doesn't allow for procedural negative pressure work like we're working on, or the helmet kind of thing, which is a negative pressure, personalized negative pressure thing. So they needed to be able to transport patients through the hospital, but it's pretty much limited to just that.
Gotcha. It sounded sort of like they were cobbling it together out of pipe from what I could hear. It's also only for use within the military, a couple of limited military contexts. So
Right.
I appreciate that, Ken. Thanks for that. Dan Hathorn, you're live.
We good?
Yes, you're fine.
Awesome. Yeah, I just wanted to throw out to the group, this has been very beneficial, and first time dialing into this, to your podcast here. This is amazing. I'm learning a lot.
We're glad to have you.
A requirement, an ask, if you will, from the VA Medical Center. They are looking for anyone who's willing to donate cloth type masks. They have a mandate for 180 facilities across the US. And there's stuff up there in Michigan as well. So it just came to me while we were on this show here, and they just asked, hey, if you know anybody who's willing to donate cloth type masks for visitors to patients inside the VA.
Gotcha.
So they're requiring visitors to patients in the VA system to wear just a simple cloth mask. So I just wanted to throw that out. Thank you.
Gotcha. Dan, stay on for just a second. I want to see if we got some of our regular callers here, because two thoughts that just popped into my head here. One is, are they insisting on cloth type, or would they be okay with the dust mask type? That's the disposable dust mask type. Because I think what we're starting to see. Oh, can you hear me?
Can hear you.
You cannot hear me?
No, sir.
Okay, well, maybe we'll follow up afterwards. But I guess I would ask if any of our textile folks are on the line. I know we have a group that does industrial sewing, and has offered it multiple times to get connected to things like that. So if afterwards we can get connected, I think Dan's ask there for the VA it's an important one, and maybe we can get connected either some of the disposable mask manufacturers or the industrial textile folks could get connected with Dan afterwards. That would be just amazing. One more great connection to make out of this. And we're so thankful to hear these connections that are getting made. We see them every week, and we hear about them every week, and it just means a lot that the community's coming together and helping each other out. So with thanks to all of you for making it happen. We really appreciate that. Okay, we're going to do last call for questions, comments, open forum. If you have a need or an effort that you're undertaking that you want to share. I see Frank Nee, you are live.
Hello, thank you, Jim, for the opportunity. Actually, I have been in the meeting every time. This is a great meeting. I actually doing some important business for Chinese company called Nutrition. They are a leading nanoparticle manufacturer in China. They have a very unique product right now. It's called the chlorine dioxide slow releasing gel. And it has been just developed in China about 3 months ago, and it has been a big market hit right now. And schools are ordering like 10,000 of those gels for the school reopening, and also other office opening settings. And it has been used in hospitals in Wuhan. It's very interesting that this slow releasing agent can really works quite well because the chloride dioxide is very highly effective oxidizing material that can kill germs 99.9%, and it's slowly releasing about last 1 month or so. So you have only a small, like 100 gram of the gels can cover about 100 square feet of space, and and it's quite effective from people's experience, and the distribution releasing rate is about 0.003 ppm. That is well below the national safety standard, and it's very effective to the germs too. So national standard is right now for the OSA is about 0.1 ppm. So we keep on looking at this. We think it can be interesting if we can bring this new product to US, because it's help people back to work and back to school, get to a normal life. And this product also has a very limited factor. It has a very short life, shelf life. So you cannot basically transport it overseas. So we are thinking about if there's a good response and the regulator approval, we can introduce this product to US here. And well, Touchstar is basically an incubator and an investment firm. This company has been early incubated from Tsinghua University Science Park. That's where we're originated from. So we have a very good relationship and bringing all kinds of products. Have this the mask that can help you prevent the germ get on top of it and has an antibiotic effect, and also other products. But this one is particularly interesting. I want to.
So this would be used as like as a surface disinfectant. Is that the primary usage?
It's an airborne air released particle. So it's primarily can fill the air with certain ppm of chloride dioxide.
Oh, I see.
And it also is effect on the surface too. I have several paper that shows it's actually kills the surface germs too.
Interesting. I think chlorine dioxide is what they put in. They treat drinking water with. That's very interesting, Frank. How can folks get in touch with you if they want to follow up and are interested in this?
Sure, I can share my email, and also I can share one very interesting YouTube that tells about the story. Actually, Chinese official media has already reported that. The Chinese Central media report about this project.
Feel free to stick it in the chat there. And is there a website that you'd like to mention verbally?
Sure. Our website, actually, we sell a lot of products through our website called teststar1market.com. So if anybody interested in getting in contact with me, definitely I can share the email, but also the website also I will put over there.
Okay, well, Frank, we appreciate hearing from you, and do appreciate your participation in all of these calls. It's good to hear from you finally. So thanks for popping in. Great. And with that, I think we've come to the end of our questions and open forum. I thank you all for being here today. This was the 10th episode of COVID Connect, and we're so grateful for what this community has done, and as well, we all work together on reopening. Can't wait to see where this goes, and we look forward to continuing the discussion with you next Friday at 2 p.m. Thanks again.